So, welcome everyone from my side. And can you hear me? Okay. So, thank you, Sonia, for giving me this opportunity to present my work here. So, I'm Ankit, and currently I published uh, this work. And today I am going to tell you about the story how we analyze special transcriptomics integrating with the single cell RNA-seq to understand the cell state covariate. So, as you might have following this RNA transcriptomics field, so these technologies are growing very fast. And couple of maybe decades ago, we had bulk RNA-seq, which measure the transcriptome of the cells uh, in a bulk. So you get the average signal from the cells in a given tissue. Then we had a single cell RNA sequencing. So each logo is a kind of thing you can think of as a cell, and which you measure from the experiment, the RNA value, and from the clustering algorithm, we define into some cell state, cell types, and based on the differential gene expression, we define every cluster as some cell type. And then we had a special transcriptomics, and this logo can be put into their position where they belong in the tissue. And in the special transcriptomics, we measure the genes where they belong in the tissue. And we had actual tissue, which is very much in 3D space. So these are the technologies which improving, and as such technologies are improving, we have a more better and better understanding of how the cells communicate in the tissue. So as a very nice review paper, it is came from the Suzanne and Julie Therot group. So she described that this holistic cell represent can be defined as like a four block spring models, where cell morphology is the one of the key block, where how the cells look like, and the cell function, which is another key component. For example, what cells do, like whether cells be described as a migration, or it's a proliferating, or it has some bad phenotypes, or in molecular sensors, what the molecule express, like transcriptome, proteome, chromatin, metabolome, etc., and of course cell environment. And this all these four components, they represent a holistic cell. And one of the goal of this all the single cell technologies, which is coming from many years, to understand this holistic cell state, or what is the holistic cell state representation. So these technologies, either it can capture a single cell ataxic, where the chromatin is open in the nuclei. Single cell cut and take, it capture the binding of DNA and the protein. Single cell RNA-seq, it capture the what are the RNA is present in the cytoplasm, and site-seq, it capture the binding of the protein in the cell surface. And every experiment, it gives some kind of output reading, and this output reading as a, like a cell matrix file, or some kind of binding component. And this can be used to understand what is the how the behavior of the cell is coming from in a given measurement. And these cells can be represented as this heterogeneity. So of course, this is only one cell cannot give you any statistics. So you have to measure this component from the many cells, and this many cells give you some statistics about in the cellular scale. We can understand what is how the cell state and their transition is move happen in the tissue space. In the molecular scale, we can understand how the genes crosstalk in a given cell or between the cells. So this can be considered as a gene regulatory network. Or in the niche scale, how the interaction is happening between the cell type, or in the tissue scale, where you have a healthy tissue, you have a diseased tissue. and what is the interaction between different sites, how the tissue become the malignant, etc. This can be understand from the tissue scale. So this talk is happening between these two scale. I will say maybe three scale, molecular scale to niche scale, which we are covering. So overall cell state from the transcriptomics measurement, it can be defined into the intrinsic variability and the extrinsic variability. The intrinsic variability, it comes from the transcription factor binding site to the chromatin, where transcription factor on and off it bind and give a gene expression noise signal. And while other side we have a extrinsic source, which is the signal, it is coming from the surrounding environment. So to understand overall cellular state variability, we need to combine these two signal, and to after combining these two signal, we can understand overall cell state variability, and we can understand the cell-cell communication. So, because this is coming from the outside of the cells, so for that we need a special transcriptomics data to understand how this overall surrounding environmental signal influencing the signal to the my central cell. So this is one of the special transcriptomics data which measure the genes gene in the every cells, and this gene has some transcript, and this each dot here is denoted as a transcript. So in this method, our goal is to understand when the two cells, I will say two cell state, when they are co-localized together in a tissue, then how the gene A, which is expressed here, how the gene B, which is expressed here, how they are correlate, what is the relationship between them. This we wanted to understand from this special transcriptomics. And one more thing, this the difference between the special transcriptomics and the single cell RNA sequencing is that the special transcriptomics with current technology, it cannot measure the whole gene expression, so it only measure only 400 to 500 genes. So based on this 400 to 500 genes, it's difficult to define all the cell type identity. So for that, we developed this tool which we call NICO, niche covariation. So in this tool, it has three modules. So first module, it does the cell type annotation. So we have some from the experiment, we have a blank tissue slide, and from the tissue slide, we have all the cells, and from all the cells, we also have all the gene expression in a given cell, and from the gene expression we want to predict what are the identity of the cell. So for this we use so this is the 2U map. So this U map is from the spatial modality, and this is from the single cell RNA-seq modality. So with this goal, we use the label transfer approach to transfer the cell type annotation from single cell RNA sequencing to the spatial. And for this, we use mutual nearest neighbor approach, and this approach find the neighbors in the two different modality. And once we find all the anchor points, then we prune it, and then we can get this kind of cell type annotate in all the cells. So this is the first module. In the second module in the program, we ask, okay, how the neighborhood can predict the identity to my central cell. So that means if I have all the cells in the this tissue, uh, so can this neighborhood predict the identity of my this central cell? So for this, I compute the for every cells whichever present, what is the neighborhood, and from the neighborhood all the cell types in the neighborhood, and from there I put in the classifier, and this classifier, it will tell me give me a confusion matrix, and from the confusion matrix we can find out okay, this is my cell types, and this cell types is predicted by neighborhood cell types, and the 1 2 which is a higher positive score, that means these two are most likely interact with the this my current interacting cell type. And based on this y-axis we can put some cutoff, and we can get this niche interaction map. So niche we call it because in a given tissue region, particular cell type which we frequently interact with each other. So then the third module, it comes the niche covariation. So when two cell types, red and the blue, co-localize together in many places in the tissue, then our question is question is how the gene I in the red one, gene J in the blue one, is what is the relationship between them. A simple relationship can be a linear regression, but in an ideal scenario where gene can be a 20,000, cell type can be a 20, this covariation lead to 76 billion pairs, which is not possible to understand anything. So for that, we use dimensionality reduction method. So one approach is the non-negative matrix factorization. For this, you can understand from this cartoon example, you have a given matrix A, and it can be decomposed into two sub-factor matrix. One factor matrix, it tell you the importance of feature in the input image, and the another sub matrix tell the facial feature set. And when you get multiply, then you get the original constructed image, which is equivalent to input image. So same idea we did it with the NMF, but this because we, so problem here that the spatial transcriptomics only measure the 400 genes, and we want to analyze all the genes. So for that we integrated along with the already available single cell RNA-seq atlas. So we take RNA-seq atlas, and this is the one reference atlas, and this is the same cell type which is present in the sequencing and the spatial, and we pull out these two matrix, M gene by N1 cell, M gene by N2 cell, and then we did integrated NMF. So this is a different version of NMF algorithm which is proposed in this paper. And this integrated version, it return the three sub matrix. So one is the W, which is common between the two modality, another sub matrix which is H, which tell the importance of feature in the cell space in the sequencing modality, importance of cell in the spatial modality, and the spatial modality cell which already has the coordinate location. So from there we learn the uh, factors, and we did the regression. So same regression we did it here in the neighborhood, and from here we can learn which latent factor of a cell is interacting with the latent factor of a neighborhood cell. So for this, if the particular score is high, so the circle size represent the p-value. So if it is high, that means this interaction is the important. And from the W matrix, we can learn all the program, whatever genes are popping up, and we can understand which are the genes are activated, and from there we can learn a lot of things. And then in the community, so biologists try to understand everything through the ligand and receptor, which ligand is communicating with which receptor when the two cells crosstalk with each other. So here when we find this two interaction is very strong, then we also uh, find this ligand and receptor enrichment when these two cell types in the gene program they interact. So from there we can, okay, this ligand and this gene program is interacting with this receptor with this gene program in this cell type. So this is our algorithm, and we tested with the many tissue, and every tissue we found something else. And but for also we wanted to test in the simulated data how it predict the niche from the neighborhood. So here I did this a very simple molecular dynamics simulation. So here I define this six cell types, and each one with the 200, and I use the interaction between T0, T2 as like a 3 epsilon potential, and T1 to T3 8, T2 to T3 10, and then I get this output, which is two-dimensional local co-localization of all the particles. And from this particle I put in my machine learning method, and from there I predict what is the confusion table. And from this confusion table I created this scoring matrix. And if we focus on this T3 niche, and T3 niche is predict has the highest score by in the neighborhood is T2, second highest is T1, and third highest is T5, and which is same as I reflected in the potential. T2, T3 has the highest potential, which is come out. So that means this niche prediction task, it worked nicely. And then we tested with the different tissue, and this tissue comes from different experiment, the different spatial technology. So in the intestine, it comes from the Murphy's technology, where we found that the crosstalk between the stem cell and the pinnate cell, and the pinnate cell when differentiated from the stem cell, then they also involved in the some Wnt pathway signaling, which maintain the stemness of the stem cell. From the mouse embryogenesis data, that is embryonic 8.5, we found interaction between cardiomyocyte and the pharyngeal mesoderm, which is help in the generation of cardiomyocyte niche in the later on. This is slide seq version 2 mouse cerebellum data. So here we found the interaction between the Purkinje neuron and the Bergman glial cell, and the calcium signaling plays some important role. And the last one is the mouse liver data. Maybe I will go in the more detail to explain what we found it. So as a general description, what is the liver tissue look like? So liver tissue organized in the hexagonal lobule, and this is the central lobule architecture, and surrounded by the portal lobule. And this portal lobule, the central lobule, lots of oxygen and the nutrient supply it happens. And in between we have the all the hepatocytes and lot of other cell like stellate cell, Kupffer cell, other cell. And this every cell types has some role in the liver regeneration and maintenance the population, or maintenance the function of the liver. So one such cell type, which is called the hepatic stellate cell, which in the normal liver it generally quiescent, but in the injury case where fatty liver acid disease happen, this stellate cell become active, and it generate lot of ECM related product, and the tissue can become fibrosis. And in this process, this TGF beta pathway, it involved in the inflammation, which plays some significant role. So from our liver tissue spatial transcriptomics, which is publicly available, we wanted to learn what new biology we can learn. So here, this is the cell type annotation module we applied, and we get a nice all the architecture of the tissue. So this is red one, which is consists of central hepatocyte, which is surrounded by portal central vein endothelial cell, and the other blue one is the portal hepatocyte, which is surrounded to these veins, and this is surrounded by portal vein endothelial cell. So visually it look nice. And we also tested with the other available methods. So for example, Uniport, 10 gram cell to location, and TACO. And this is the Jacquard similarity, which is compute the intersection over union. And for different methods, we get the almost a very good accurate score for the different tissue. So then next we applied the neighborhood analysis, and from the neighborhood analysis we get a nice score of this central vein architecture, which is consist of this zone, and the portal vein, which is make up this architecture, and the central zone, which is consist of central hepatocyte, central vein, which is zone present in this zone. And one of the interaction we found the stellate cell interacting with the Kupffer cell. And then from the covariation, we ask, okay, what is the gene program is activated between factor 2 of stellate cell, factor 1, which we found significant. And then we look in the genes, what are the genes are activated. And from the gene, we understood that the some of the immune related pathway in immune related genes are active in the Kupffer cell, and in the stellate cell most ECM related genes are activated. Then the question is why these two gene covariates are connecting. And from the this ligand receptor analysis, we found this TGF beta ligand in the Kupffer cell is interacting with the TGF beta receptor in the stellate cell. And from our hypothesis, then we propose that when the bacteria or infection it come, then this Kupffer cell, which is also called macrophage, it sense this bacteria and activate this hepatic stellate cell pathway through TGF beta, and they repaired it. But in the chronic infection where this keep on infection is happening, this pathway become insufficient, and this decoherine and glycan can regenerate to fibrosis. So to test this hypothesis, we play different.
Ankit, just to let you know, you have about maybe 3-4 minutes.
Okay,
so
okay,
you know,
it will end. Yeah, yeah.
No problem. Thanks.
Yeah, yeah. So we wanted to test this hypothesis, how the TGF beta concentration with the decoherine concentration, which block this pathway, how we can understand. So this is a in vitro culture, cell culture experiment performed by my colleagues. And this is the qPCR reading, and this qPCR reading ask the expression of this activated stellate cell marker, which is denoted as each time point, and the different concentration of TGF beta and the decoherine, which show as a significant p value. So this suggests that when the decoherine concentration is put into this, then TGF beta pathways signal get reduced. So this is, and also from the this is a single molecule phase experiment, we showed the similar things where we found the decoherine signal in the stellate cell, which co-localized with the Kupffer cell with TGF beta. We have a correlation of 0.59. So this suggests that this covariation and cell-cell communication, it can find some interesting biology. And one of the recent uh, review paper which suggests that this kind of task is very important in the tumor field, in the cancer field, where lots of question is like what is the cell identity, what is the cell state, how the cell state interact, and this can be applicable. So in this NICO methods did the cellular neighborhood analysis, cell type annotation, cell type interaction, ligand receptor, etc. And it will be very useful for the disease tissue where really some of the cells are has a bad phenotype, and if we can understand which are the genes are involved in this bad phenotype, we can understand and we can try to find the therapeutic site. So this was talk I presented, but I also involved in the in spatial biology, but not including the molecules. So if you want to know these two story, I'm happy to talk some other times. And thank you. I'm happy to take any questions.